Here is my DNA model. I promised you guys I'd show you guys more in depth. So yeah, we're gonna play around with this today. So let's get into it. Transcription and translation. This is the second lesson in Unit 10, which is protein synthesis, aka central dogma. Let's get into it. So our lesson objective, let me make myself a little bit smaller to show you this graphic that I made. So this is the cell. Inside of the cell, we have multiple organelles, but today we're just focusing on two main ones. We're focusing on the nucleus. The nucleus's job is to protect DNA because DNA is extremely important, right? It gives the instructions to do everything in the cell, everything in the body. So DNA is very important. It needs to be trapped and protected in the nucleus. Okay. However, DNA's information, a copy of the DNA, must be sent out of the nucleus and into the ribosomes, which are known as the protein factory. That's where proteins are made. And we should all already know that proteins do all of the jobs in the body. So for example, there's a protein called keratin. Depending on how keratin makes its sheets, that'll tell if your hair is going to be straight, curly, or wavy, or even kinky curly like mine is. Proteins also such as insulin, an enzyme, insulin, controls like how your body processes sugar. So if you're insulin deficient, you have one of the different, then you may be a diabetic person. Okay, so again, your DNA controls the proteins that are made, and the proteins are actually what gives you your traits, your characteristics, what makes you you. Okay. So just to recap, central dogma, the law of life. So of course we start off with this thing called DNA. It's double-stranded. It's protected in the nucleus. DNA has multiple parts. I'm going to unwind it just so I can show you. DNA has multiple parts. It has a sugar phosphate, also known as the sugar phosphate backbone. Let me actually make it bigger. So DNA has multiple parts. So again, let me just show you again. I can't show you enough because this is very important in biology. So this is DNA. When it's very, very tightly and condensed, it's what's known as a chromosome. We'll talk about chromosomes a lot in the next unit. So it's also known as a chromosome. However, sometimes DNA can be unwound like this, okay? And that's whenever you can see its individual pieces. So it has a sugar phosphate, which is also known as a sugar phosphate backbone. That is what keeps DNA stable. And then what actually makes each person's DNA different from each other is the order of the nitrogenous bases, the order of the bases. So we have adenine, thymine, cytosine, and guanine in DNA. So A, T, C, and G. And also one more thing about DNA, one phosphate, and one of the bases is a small unit. That is a monomer of the entire DNA molecule. That monomer of the nucleic acid is known as a nucleotide. Okay, lots of vocab here, lots of vocab here. Hopefully my repetition is going to help it stick in your brain, but if not, I also have the Quizlet cards, so make sure that you're also paying attention to those and using those on your work. Okay, DNA is copied into RNA, which is a single strand of nucleic acid. RNA is not super important, so it can leave the nucleus and go to the cytoplasm. In the cytoplasm, it's going to find the ribosome, and on the ribosome, we're going to assemble a protein. Proteins are assembled out of using individual amino acids that are put together like beads on a string. So you can think of it kind of like a bracelet. Whenever you have a beaded bracelet, right, you have each individual bead that works together to make the entire bracelet. Well, think of each individual bead on a bracelet or on a necklace as amino acids, and think of the entire necklace or the entire bracelet as the protein. Okay, and again, proteins give us our traits such as hair color, height, weight, so many different things. Okay, again, DNA gives us our unique identity because we have that sugar phosphate backbone that is conserved between all organisms. Every single organism on Earth has a sugar phosphate backbone. What makes us different, what makes us unique, is the order of the A, C, T's, and G's. That order, also known as the sequence, is going to determine what amino acids are made, and our amino acids determine how our proteins are folded. Our proteins folding determines our actual traits. So again, this is a recap. That's why I'm just kind of going through it quickly. Here it is once again. I'm trying to show you. Notice I'm showing you in many different ways because I really want you guys to stick this information into your brain and keep it there. So again, DNA has to stay protected inside of the nucleus. So what we do, we have this process called transcription. Transcription turns DNA into RNA. RNA is able to leave the nucleus. It's going to carry DNA's message from the nucleus into the ribosomes. Once it's inside the ribosomes, RNA is going to get translated into the language of the protein. Proteins are made on ribosomes, and again, we know proteins control cell functions. And then there's this process, it's called gene expression. So some genes are turned on and some genes are turned off, and that's what gives us our traits. We'll talk more in depth about gene expression and chromosomes and all of that in coming lessons. So let's do a quick check for understanding. DNA is located in the blank. Where's DNA located? Can DNA leave this organelle? And what is DNA's function? DNA's function is to make what? Okay, so the answer is DNA is located in the nucleus. DNA cannot leave the nucleus. And DNA's function, you could have said DNA's function is to make mRNA. DNA's function is to make proteins. Those are, those are all acceptable answers. Okay, another check for understanding in DNA. So in DNA, if I have an A nitrogenous base here, if I have an adenine here, what must go on this side? What matches up with adenine? How about this? If I have a G right here, what must be on this side? Okay, good. How about RNA? What are my RNA binding, my complementary bonding rules for RNA? All right, good. So in DNA, we have apples on trees, cars in the garage. In RNA, we have A, but A doesn't bond with T anymore in RNA. A is going to bond with U, right? We don't have T's in RNA. And then G still bonds with C. So let's go ahead and compare DNA and RNA before we get into the specifics of transcription and translation. Let's just check for understanding. So this right here is a Venn diagram. Hopefully everyone knows what a Venn diagram is. So basically, if it applies to DNA, it's going to go in DNA circle. If it applies to RNA, it's going to go in RNA circle. And if it applies to both of them, it's going to go in that overlapping part. So here's our word bank. So we're going to sort each of these things individually together. So let's do this first example. So DNA or RNA leaves the nucleus and goes to the cytoplasm and ribosomes. Will that apply to DNA only, RNA only, or to both? Going to be RNA only. RNA can leave the nucleus. DNA cannot leave the nucleus. Okay, so we're done with that one. The next one we're going to do is this one that's highlighted, that's outlined. C bonds with G. Where would that go? Where would C bonds with G go? DNA, RNA, or both? Both. C bonds with G in both. Next up, we're going to have the nucleotide. So the phosphate, the sugar group, and the organic base. Nucleotides. Is that just DNA? Is that just RNA? Or is that both? It's for both of them. All nucleic acid monomers are the nucleotide. Next, A bonds with T. Where would that go? Good, DNA. A bonds with T. Apples on trees. A bonds with U. Where would that go? Good job. Definitely for RNA. A bonds with U. Next up, we have stays in the nucleus. Is that DNA, RNA, or both? DNA only. DNA must stay in the nucleus. RNA can leave the nucleus. Good job. We're almost finished, almost at the end. How about this? One thing to note about this is that it is single-stranded. And you see that it's single-stranded because it has one sugar phosphate backbone and all of its bases are unpaired. RNA is single-stranded, exactly. Last but not least, the double helix. Two strands, definitely DNA, double-stranded DNA. Perfect. Okay. So again, now we're going to get into the specifics, some more things that you have to know. So DNA into RNA is known as transcription. So that's sending DNA's message out of the nucleus. It's going to transcribe half of the DNA into an RNA. Okay. RNA to protein is what's known as translation. That's actually turning DNA's message into a functioning protein. Okay, so some more checks for understanding. Where is DNA located? Notice that I'm asking you this over and over again because it's very important. Where is DNA located in the cell? Can it ever leave? DNA is located in the nucleus and it cannot leave. It can't leave because it is too delicate. It's too precious. It's too important and needs to be protected in the nucleus. Okay. Where are proteins made? And how does DNA's information get there? Proteins are made on the ribosomes. That's the protein factory, is the ribosome. And how does DNA's information get there? This arrow is representing the movement of mRNA, messenger RNA. Okay, here's the same information, just in a different representation. So this right here is DNA. I have a question though. How do you know this is DNA? Obviously it says it right there, but how do you know this is DNA? Okay, so there's two things that you could have said. There's T's. We know that T's are only found in DNA. The other thing that you could have said is that it's double-stranded. We have two strands that are bonded together. That's DNA. Okay? The process of turning DNA into mRNA is known as transcription. You're transcribing from one nucleic acid to another nucleic acid. That's transcribing. Then translating is going from the language of the nucleic acid to the protein, right? Because remember, we have our four biomolecules: carbohydrates, we have carbs, we have lipids, we have nucleic acids, and we have proteins. So nucleic acid is one language. Protein is another language. So from going from nucleic acid to protein is a translation. So again, transcription is transcribing DNA into mRNA, going from a language of deoxyribonucleic acid into messenger ribonucleic acid. So they're still nucleic acid, so you're staying in the same language, you're just transcribing. While translating would be changing languages. So you're going to translate messenger ribonucleic acid into the language of protein, which is the amino acid. It's one way to, because I know like when I was in high school biology, I was very confused on like transcription, translation. Transcription happens first, translation happens second. And also, again, transcription is going from the same language, while translation is going from one language, nucleic acid, into another language, the protein. Okay. So again, you can go ahead and read this, but this is the exact same thing that we had last time. So essentially, all it is that mRNA is being read by the ribosome. The ribosome then attracts a tRNA that has an amino acid, and they're being put like beads on a string to make the protein. Okay, more vocabulary. Codon. A codon is just a 3 nucleotide sequence of mRNA. So, for example, if this mRNA sequence is AUG, ACA, AUG is a codon. Then the next codon are the next 3 letters, which are ACA. Okay. Also remember that a codon is going to match up. So every 3 letters is going to match up with an anticodon on a tRNA. That tRNA is going to carry a single amino acid. Okay. Here's what it looks like in just yet another representation. So again, we have the mRNA. We have 3 bases read at one time. Those 3 bases are known as the codon. The codon is going to match up with an anticodon. So notice that anti means just the opposite. So where there's an A on the codon, there's going to be a U. Where there's a U on the codon, there's going to be an A on the anticodon. There's a G and a C. So they're the same pairs. We already know RNA pairings, A, U, C, G. There's also this tRNA. Again, the tRNA has the anticodon, and the tRNA has the amino acid attached to it. Okay, we're going to look at some animations of this in the next lesson. If this is not making complete sense to you right now, don't worry. Like you'll when you see it in action, you'll be like, oh, okay, that's how it happens. Okay, same information, just in a different way. 3 nucleotides is 1 codon, which is 1 amino acid. So, for example, if this is the mRNA sequence, the mRNA codons just means that there's 3 nucleotides. So AAA, CGU, UCG. Okay. Amino acids, every 3 codons makes 1 amino acid. However, how did I know what codon codes for which amino acid? It's easy. You're going to use a codon wheel or a codon chart. So this is what it looks like. This is what a codon wheel, codon chart looks like. Okay, you might have seen this before, maybe you didn't. And basically, just like in baseball, we're going to always start at the first base, determine what letter. Then we're going to go to the second base, determine which letter. We're going to find out where those two match up, and then we're going to look over here at the third base and see which one it is. Okay, if that doesn't make sense, we're about to do it. We're about to do it. We're about to do it. Don't worry. So let's find the mRNA sequence ACG. So again, in baseball, we're going to start with first base, then second base, then third base. So let's look at our bases, our nitrogenous bases. First is A, second is C, third is G. Boom. Now let's start. First base is A. Let's go over here where it says where we see first base. First base is A. So I know I'm going to be somewhere along here. Okay. My second base is C. So let's go up here to second base. C, bring our hand down to where A was at. So this is where A and C meet up, right? We see that. Everyone can see that's where A and C are. Now it's one of these 4. How do I know which 4? I'm going to look at my third base. Third base says G, so I'm going to come over here. Third base, find G, come over here. Boom. That's it right there. A. So it's somewhere in here. C. That means it's definitely in this box. And then G. It's definitely this one right here. Okay, next sequence we're going to find out together. G, G, G. So my first base is G. So I know I'm somewhere down here. My second base is also G. So we're going to come here to G. So I know G and G meet up here. My third base is G. So that's going to be glycine. Glycine is my answer. Okay. Next, UAG. First base, U. Second base, A. U and A meet up in this box, one of these 4. Third base is G. So U, A, and G stop. So the answer to this would be stop. Okay. Yeah. I want you guys now to find out for me. I'm gonna make one up. UGC. Y'all will find for me UGC. Okay. First base is U. Somewhere in here. Second base is G. Somewhere in here. Third base is C. Cysteine. Cysteine. Boom. That simple. It's that easy. Okay. One thing that I do want to know, I know I keep going back. One thing that I do want to know is that please make sure that you are using your fingers to do this. Because if you're just looking like da 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 da, you're going to mess it up. I promise you. I've taught this before. You're going to mess it up if you're not putting your fingers on the screen and actually dragging them and doing basically what I'm doing with this. You should be doing that with your fingers. Okay, that's the best way to not mess this up. These problems are too easy to take an L on. Like if you take an L on this, no, there's no need to do that. All it is reading the chart and taking your time and doing it. Okay, the VIP codons are going to be AUG, which is the start codon, aka methionine. UAA, UAG, and UGA are the stop codons. You don't have to memorize them because they're on the chart, but just know that that's AUG is always where the mRNA, the protein transcription is going to begin, and UAA, UAG, and UGA are always going to be the stop codons. Okay, they're just VIPs. Yeah. So here's just some more representations of the same thing we've been talking about. We have the mRNA, what it looks like, and then these are the letters. We know we break them up into groups of 3 for the codons, and we use the codon wheel to find the actual names. One thing to note is that oftentimes the amino acid names are long, so we will abbreviate them. So ARG will be for arginine, SER for serine, GLU for glutamate, THR for threonine. Okay, don't be confused on that. It's just going to be the first 3 letters of it. Okay, here's another representation of the exact same thing. This is an mRNA. How do I know that? Well, it is single-stranded. I also know that because there are U's in it, and our codons are how many letters? 3 letters. Okay, this is our mRNA. And as we can see, this is the codon that they choose to highlight in this picture, UCG. The anticodon then will be U matches up with A, C matches up with G, G matches up with C, so that's the anticodon. The anticodon is on the tRNA. As you can see, it even looks like a T. You can see it looks like a T. That's the tRNA, the transfer RNA. Its job is to transfer this amino acid onto the growing polypeptide, aka protein chain. Well, again, another representation of the same concept, mRNA, codon, and the tRNA carrying this amino acid. Okay, let's do some more practice together. So essentially, if I give you the mRNA sequence, then you should be able to also find me the DNA sequence, right? Because we know the pairing rules. So we're just going to use ATCG. So if the mRNA codon is AAA, what is the DNA sequence for that part? TTT, right? So we have AT pairs and CG pairs. All right, how about the next one? Do it on your own. Good job. G, C, A. How about this last one? U pairs up with A, C pairs up with G, G pairs up with C. Okay, and I know the A's and the U's and the T's can be a little bit confusing, so let me just tell you this. You always just double check yourself. So let's say if I was confused on if I should have put a T or an A there. I'm not sure. Pick one, do it, and then check yourself by going backwards. If I put an A up here, if I go backwards, if I'm going from DNA to RNA and I'm doing, and whatever pairs up with A, would I write a T here? No, because RNA doesn't have a T. RNA has U's. That way I know my answer is correct. So always just check your answers. So here is the summary of everything we've just learned. So DNA sequence ATCG, mRNA AUCG, tRNA AUCG. RNA has the same rules. Amino acids, we're going to use the mRNA sequence on the codon chart. Okay, so for example, if I have a DNA sequence that is TTT, converting that to RNA is going to be AAA. Then converting that into the tRNA anticodon is UUU. In order to find the amino acid, I'm always going to use my RNA. One thing that students get mixed up on whenever we throw in this tRNA in here is that y'all sometimes try and use tRNA to find the amino acids. That's wrong. You can't do that. You have to use mRNA. We already know mRNA is what attracts the tRNA. Okay, so again, you should be able to go DNA to DNA, right? So ATCG. You should be able to go DNA to mRNA, AUCG. You should be able to go from mRNA to amino acids using the codon chart or codon wheel. You also should be able to do mRNA to the anticodon. Okay. You also have to be able to do any of this backwards as well. Backwards, forwards, side to side. It's not confusing though. We're going to practice it together. So I can give you a problem like this and I ask you, fill in all the missing squares. And you're like, but there's so much missing. How can I do this? It's really not hard. Let's do it together. So let's just take it one column at a time. One column at a time. So if we have a DNA sequence that is TTG, what will be the mRNA? Well, T pairs up with A. T pairs up with A. G pairs up with C. AAC. Okay? Now, I always go mRNA and then to amino acid just so that I don't accidentally do the tRNA amino acid. So mRNA, find the amino acid. Using a codon chart, I have one right here. You can just Google one. They're all the same. Aspirin. Okay? And then for our tRNA anticodon, we're just going to do A pairs up with U, A pairs up with U, C pairs up with G. UUG. Boom. Just like that, we filled in all of that by we were given 3 letters and then we found out all this other information. What's amazing is that your cells do this in like the blink of an eye all the time, millions of times a day. Okay, now let's work backwards. We're given the amino acid. Let's work backwards to find the mRNA, the DNA, and the tRNA anticodon. So let's take your codon chart. We're going to find glutamine. And on the so on the codon chart, we're going to find GLU. And you'll see that there actually are 2 different ones. We could do CAA or we could do CAG. So since this problem doesn't give you any other information, you can just pick one. Okay, it doesn't give you any other information. You just pick one of the 2. So I just picked CAG. If you pick CAA, whatever, just make sure that your other information is correct. Okay, so from CAG, how do we go backwards into the DNA? G, T, C. Okay, how do we go from mRNA to the tRNA? Well, we're going to do G. What's next? Good. GUC. Okay. Last but not least, you're going to do these on your own using your codon chart and your base pairing rules. So I'll give you some time to do that. Good. Check your answers. Make sure you're good. All right. So that is the end of that. So transcription translation, pretty simple topics. Just make sure that you don't think that they're so simple that you're messing up your A's and your U's and your T's and all of that. Make sure that you're taking your time. Or what I like to do whenever I have a test or a quiz or something, I like to just like write down the base pairing rules in the corner real quick when I first get it. That way I always just refer back to that and then I never will get it wrong. Test taking skills, how to finesse the system. Make sure that you know how to finesse the system. Okay? Go ahead and get onto your exit ticket and I will see you guys later. Bye.